So notice first thing it says that they showed life could be created in a test tube. I really want you to know he did not create life. He just showed that inorganic molecules could turn into organic molecules over time. So and this is the answer. This is the correct answer. So this is the proof. Miller experiment is the proof that we have that life was possible over time, and humans didn't need to be around in order for this to happen, where inorganic molecules just turned into organic molecules. So this is very important, so I just want to write it out for you. So there are two parts of the puzzle. One is organic molecules. So inorganic molecules turned into organic molecules. That's the first part that happened on early Earth. What is the second part? You guys want to help me with this?
Molecules stick with the clay, and the clay have what amino acids stick together.
Yeah. So with the use of clay, genetics happened. Genetics happened, and molecules such as RNA turned into self-replicating molecules. So this is how genetics might have happened. So that's the second part. So self-replicating RNA came around with the use of clay. So just try to remember these two parts, and this happened with the Miller's experiment. So this is very famous experiment. That's the one that they are using to show the evidence for inorganic molecules to turning into organic molecules. Okay, so next thing we have is we're gonna go over how membranes enclosed genetic material. So this is first cell, and I already said that first cell is called protocells. It's like a prototype, first thing that came around. This was very, very, very simple. This is lipid membranes. They came together, and it separated the outside environment from inside. And then inside you have self-replicating DNA, RNA first, that's the one that came around, and then the proteins. So this is what gave rise to the first life. These simple cells later turned into prokaryotes. So protocells are the first cells that came around that was very simple. It only had lipid membrane, genetic material such as RNA and proteins. And later they turned into more complex prokaryotic cells. This lipid membrane, the whole purpose for this is to separate the outside environment from the inside. Once that's different, then more complex things can come around. So prokaryote is a single cell organism that lacks a nucleus. Pro means before, karyo means nucleus. This is before nucleus. So prokaryotes, they lack nucleus. Prokaryote is a domain. Another domain we have is eukarya. Under prokaryotes, you have bacteria and archaea. So you have under prokaryotes, you have two groups, bacteria and archaea. Prokaryotes, both of these groups, they don't have a nucleus and they don't have membrane-bound organelles like eukaryotic organisms. So we have two domains here. Bacteria and archaea, they're prokaryotes that are in different domains, and they were able to put these in different domains, comparing the type of environment they live in, and also they were able to compare their DNA because we have that technology now. So just to make sure you guys are following me, I'm gonna try to keep a record of all the different groups we go over. Have prokaryotes, and they have two groups. One is bacteria and the other one is archaea. Prokaryote means no nucleus. Pro means before, karyote means nucleus, no nucleus. And also no organelles. Okay, so going back here, this is showing you an example of bacteria, and this is showing you an example of archaea. I'll tell you why they're called archaea. So what happened was when they found this group, archaea, when they started finding organisms that go in this group, they found them in very extreme environments, maybe where it was very hot or very salty, or maybe in a lot of methane gas. So they found these organisms in areas that were very similar to early Earth. And the term archaea means old. So they thought that these came first because they are living in similar environments as the early Earth had. So they named it archaea. Later, they were able to sequence the DNA for bacteria and archaea. So once they were able to sequence the DNA, they found out that bacteria came around first and archaea came after. So they already named it archaea, means old. So the name is same, but now they know that the bacteria came first and then archaea came around. So they were able to do that, distinguish that using DNA sequences and chemical compositions between to find the differences between these two domains. So I would make a flashcard for this. This is an important slide. So prokaryotic cells, we already said that it means no nucleus or other membrane-bound organelles. So we already wrote that down. The size for this is from 1 micrometer to 10 micrometers. 10 micrometers is the size of a printed period. That's the biggest prokaryotes they found. They found it in guts of a fish and also in coral reefs. So that's the biggest prokaryote they were able to find. It has circular chromosome, and they're unicellular. Unicellular means single cell. They're simple. They're single cell organisms. Some of them can fix nitrogen. So nitrogen that's in the environment, it's locked in and it has to be, converted from one form to the other, and bacteria does that. So when you're breathing about, 75% of nitrogen. You wouldn't be able to do that if this bacteria wasn't fixing that nitrogen. And then some of these prokaryotes, because I already told you some of them are archaea, they can grow at temperatures that are above 80 degrees, means they can be living in boiling water. So they can live in extreme environments. So again, you can have a question on the exam where I ask you the size for prokaryote, and you're gonna say it's between 1 to 10 micrometers. Maybe I'll give you options 1 micrometer to 10 or 10 to 100 or something like that. So just try to remember these different things about prokaryotic cells. So make a flashcard for this. So some things that are unique to bacteria only, not archaea. We're talking about only bacteria. Cell walls are composed of peptidoglycan. Anybody knows what peptidoglycan means? You guys know what peptidoglycan means?
Is it sugar proteins?
It's sugar protein. So peptide means protein and glycan means sugar. If someone is hypoglycemic, they have low sugar levels. So it's made up of some parts of protein and some parts of sugar. So I want you to remember this. There will be a question on the exam. Bacteria cell walls are made up of peptidoglycan layer. if you take microbiology, they will go over this and you'll be able to stain this cell wall, and that's how you will be able to distinguish between different bacteria species. The membrane based is on fatty acids, same things our cells are made of, is made up of phospholipid bilayer. Some use chlorophyll in photosynthesis. So if you have a fish pond and you see that blue-green stuff on that fish pond, that's cyanobacteria, and it's using chlorophyll to do photosynthesis. That's why it looks green. It cannot generate methane. This is important because archaea can generate methane. Bacteria cannot generate methane. And then it says it's sensitive to streptomycin. Do you guys know what mycin means? So this means fungus. Strep means bacteria that's in chains. So streptomycin, it means if you have some bacteria and you put antibiotics, that's called streptomycin, it's going to kill the bacteria. If you have to take microbiology class, they will give you this unknown bacteria and they will ask you, find out what type of bacteria is this. So when you try to find that out, you're gonna use maybe different antibiotics. One thing you're gonna know that it's gonna be sensitive to streptomycin. That's even to find out if it's a bacteria or archaea, because archaea are not sensitive to streptomycin, only bacteria are. So if you're looking under the microscope on bacteria and archaea, you can't really tell the difference. Then you can use the streptomycin, and if it dies, it's bacteria. If it doesn't, it's archaea. So you can use this to find out which one it is. Then it says genes do not contain introns. You guys remember what introns are? You guys remember?
Non-coding area.
Can you say that again, Truce?
It's a non-coding area.
It's non-coding area, it's non-functional. It's all that garbage DNA that has to be taken out before that DNA material moves into the cytoplasm. We covered that in transcription chapter, I believe. So they do not have this garbage DNA. They do not have introns like our DNA does. Okay, now some features that are for archaea. One thing I need you to remember, I already told you that bacteria cell wall is made up of peptidoglycan. Archaea cell wall is not made of peptidoglycan. I don't care if you remember what type of material is found in archaea, but just know that it's not made up of peptidoglycan, it's made up of something else other than peptidoglycan. Membrane is non-fatty acid lipids, and they do not use chlorophyll. They cannot do photosynthesis. They are found in extreme environments where there's a lot of salt, it's really hot, there's a lot of sulfur, there's a lot of carbon dioxide or methane. So they're found in extreme environments mostly. There are some that are found in normal temperatures. They are insensitive to streptomycin. So this is antibiotics, it's insensitive. So if you have two groups, you have bacteria and your archaea, you look at it under the microscope, you cannot tell which one is which. You can just use this antibiotics. If it doesn't die, then it's archaea. And it might have some introns. So some of these have introns, and we have introns, we are eukaryotic. So this archaea is closely related to eukaryotes compared to prokaryotes, but it's still prokaryotes because it doesn't have a nucleus, but it's closely related to eukaryotes. Any questions so far on bacteria or archaea? It's mostly memorization. Just make a flashcard and try to remember differences. Another thing I said, it generates methane. That just reminds me, I want to show you something really quick. Have you guys heard about swamp gas? So what happens is when there's a swamp, water is standing there for a long time, all the vegetation dies and it starts to decay. There is usually no oxygen. Oxygen supply depletes. So there's no oxygen. And there's archaea that starts to break down those dead leaves, all the vegetation. And then when they do that, they generate methane. So when there is methane gas and there's lightning, maybe there's lightning that happens, it can put that swamp gas on fire. So you might see these fireballs. So this is where a lot of those fairy tale stories and ghost stories come from. So that's the methane gas in swamps is coming from the archaea species that might be that might be feeding on those dead leaves. Also, I just remembered Bogman. So this is in, I think, Ireland. This person was murdered and thrown into a swamp. And because there's no oxygen there, the body was completely preserved. And you can see so much detail here, even the wrinkles are there. So the body was preserved because there is no oxygen there, no oxygen supply. Um, there are a lot of stories about different, bogmans. They're called bog mummies as well. So again, just to, I hope that helps you a little remember that the archaea can generate methane.